My name is Yiouli Ktena. I am a third-year pediatric hematology oncology fellow at the combined Hopkins NIH program, and I would like to thank you for this wonderful event and the opportunity to present our research today. I have no disclosures, and today I'm going to introduce three concepts: bone marrow transplantation, graft-versus-host disease, and DNA methylation and how it controls graft-versus-host disease, as we found out. So bone marrow transplantation, or bone marrow, you can think of as the factory where the hematopoietic stem cells are the factory workers, and those make our red blood cells, which carry around oxygen, our white blood cells, which make up our immune system, and our platelets, which help us stop bleeding. And when we talk about bone marrow transplantation, we're really talking about hematopoietic stem cell transplantation. So how does that work? We very carefully pick our donor, and we collect the stem cells, either from the bone marrow, or we can collect them from a vein, or from the cord blood when a baby is born. And then we give what is called the conditioning regimen, which is a combination of chemotherapy and perhaps radiation in some cases. And that has two purposes. Number one, to eradicate disease in patients who have a malignant disease. And number two, to make space for the new bone marrow, the new immune system. And then lastly, the last step is the infusion into the host or the recipient. And that's the least impressive step because that really just looks like a blood transfusion. But what is very impressive is that the stem cells go and find their way into the bone marrow and give you a new beautiful immune system. So hematopoietic stem cell transplantation can be autologous, where the patient's own stem cells are collected and then reinfused for some reason after therapy, or most commonly what we refer to when we talk about a BMT is an allogeneic transplantation. So we collect the stem cells from a donor, either related or unrelated. Like Dr. Shah said, despite advances with CD19 and CD22 CAR cells and other exciting things, it remains the only curative therapy for some of our patients. Patients with malignant diseases such as high-risk leukemias or lymphomas, and patients with non-malignant diseases such as bone marrow failure, immune deficiencies, or other genetic diseases. However, graft-versus-host disease continues to be a major limitation. So what is graft-versus-host disease? The way I think about it is that we very carefully pick our donor, and we match our donor and our host, and we give the graft. And the donor's T cells go in there, and they suddenly realize that, hold on, everything here looks similar, the furniture is similar, and the curtains are similar, but I'm actually not in my own home. And then they get angry and start attacking everything. So if you think about a kidney transplant where we talk about rejection, where the host rejects the kidney, here is kind of the other way around. The donor realizes that it's in a different environment and sort of rejects the host. Nearly every organ can be involved, and it can be life-threatening. And despite our best efforts to match the donor and the recipient, there is always a risk for graft-versus-host disease unless we're dealing with identical twins, where by definition, all your genes are the exact same. So the mechanism of GVHDs, you initially have your conditioning regimen, as we talked about, that causes diffuse damage by definition. And then that releases cytokines, which activate your host presenting cells. And those presenting cells activate your donor T cells that are now what we call alloreactive. You also have other inflammatory agents that further amplify injury and inflammation, and all of that results in graft-versus-host disease. So the ideal transplant, or holy grail transplant, should allow for full engraftment, so you have a beautiful new bone marrow and immune system, should have no graft-versus-host disease, but should allow for graft-versus-tumor effect, where your graft realizes that there are cancer cells that are not supposed to be there and helps eradicate them. And then we'll take a second to talk about epigenetics and DNA methylation. So epigenetic changes are changes that happen to the DNA after it has been created and don't affect the DNA sequence itself, but they affect how open or tight the DNA is. So tight DNA is hard to read, it's not accessible, but open DNA is easy to read and ready to do its job. These occur throughout development. So in an embryo, for example, genes are turned off and on through epigenetics to create a human. But they also happen throughout our lifetime in response to toxins or stress or disease. And DNA methylation is a type of epigenetic change where you have addition of methyl groups to specific areas of the DNA, and subsequently those methylated areas are silenced. So whatever is methylated is silenced. These are heritable in daughter cells, and today we're going to focus on the main enzyme that does this, which is called DNMT3A, or DNA methyltransferase 3A. So to summarize, epigenetic changes regulate gene transcription, are inheritable, and are reversible, so they make excellent therapeutic targets. Now, we've said that graft-versus-host disease depends on T cells, and we also have found out that T cells heavily depend on DNA methylation. So baby T cells, naive T cells, start out as naive, and in response to activation, acquire a new personality and a new role that is stable. So for example, T helper 1 cells are supposed to make interferon gamma. And the way they do that is that the interferon gamma locus undergoes demethylation, so therefore is open and accessible, but they're not supposed to make IL-4, for example. So that locus is methylated and silenced. And in T helper 2 cells, the opposite changes happen. And those changes now result in a stable new personality for each T cell. So our hypothesis has been that DNA methylation of donor T cells regulates alloreactivity after transplantation. And how are we testing this? We are using mouse models of graft-versus-host disease, and we are using mice that have been genetically engineered to lack DNMT3A in their T cells only. So these mice are identical to wild type or normal mice, except their T cells cannot methylate their DNA. And I'm going to talk a little bit about murine GVHD models. So we take our donors, and in this case, they are T cell wild type, DNMT3A wild type and knockout. We isolate their bone marrow cells, and we isolate splenic T cells as well, because mice do not have enough T cells in their bone marrow to cause GVHD. And then this is a haploidentical match, so that we're sure that with the half match, the recipients will get graft-versus-host disease. The conditioning is irradiation in mice, and we give the graft, the bone marrow cells provide immune reconstitution, and the T cells give GVHD. And the outcomes that we follow are survival and weight loss and a clinical GVHD score. And you can see on the right, that's a is that your left, that's a syngeneic recipient, so sort of like getting a transplant from your twin. So that mouse doesn't have GVHD and is nice and healthy, nice shiny fur, but the other mouse has got an allogenic transplant and has GVHD, so it's hunched and missing fur, and in real life, you would have to poke it to move. And I don't know if you've interacted with mice. They don't sit around for you to poke them. So what we have found is when we use the DNMT3A knockout T cells, we accelerate GVHD. So we have used our conditioning and graft as we talked about, and what we're looking at here is a Kaplan-Meier curve. So you start from survival 100%, and anytime you have a step down, that is an animal that we have lost, decreased survival. So the syngeneic donors in green do just fine, no graft-versus-host disease as you would expect, as they should. And then our wild-type group that get the transplant from the wild-type recipients get some graft-versus-host disease as they should. But then the knockout group does much, much worse. And that is accompanied by worse GVHD scores and worse weight loss. So who is the culprit? Who is to blame? When we have looked at the spleens of these animals, and what you're supposed to see in the spleen after a few days, you're supposed to see T cells come back, and they have CD4 and CD8 T cells. That's the way they're supposed to be, the two main groups, as Dr. Shah said. That's how they isolate them. What we found in the knockout T cells is this very impressive population of double positive T cells. So this population that is coming out of the CD8 population, but is also expressing CD4. And in general, those are not supposed to be there. So double positive T cells can be found in the thymus as our T cells are maturing. But once a T cell matures, it's either CD4 or CD8, and in general, that's mutually exclusive. And there have been some reports of double positive T cells in the periphery in the context of HIV or lupus, but not to this extent, and their role and function is unknown. So this is quite unique. And we have encountered this in multiple different organs at multiple different time points. So then we thought, because these CD double positive T cells seem to be originating from the CD8s, maybe this is a CD8-driven process. And our hypothesis was, if the CD8 T cells are to blame, perhaps if we mix and match the CD4 and CD8, then we can tell indeed who is to blame. So we separated CD4 and CD8 and then mix them again to co-inject them. So again, we have our syngeneic control group that's just fine, our wild type and our knockout recipients. And then this is the group that got CD4 knockout T cells, but wild type CD8. And statistically speaking, it's actually no different than the wild type group. But then you have the group that gets the knockout CD8. And that group is actually, statistically speaking, no different than the knockout group. And the two new experimental groups are different between them. So that does seem to be more of a CD8-driven process. And we have confirmed the same thing in an MHC1 mismatched model. So when you only have an MHC1 mismatch, you only have CD8 activation. And not only that, but our graft only contains CD8 T cells. And indeed, we saw the same thing. And with the CD8 only dependent process, this is even more of an impressive difference. So we tried to find out why this is happening, and we've looked at cytotoxic activity between wild type and knockout T cells. Perhaps the knockout T cells are better at killing. And we've used this method called a JAM assay, which, by the way, JAM stands for just another method. And what you do is you activate your lymphocytes, and you mix them with tumor cells that have been fed with thymidine, radioactive thymidine. So then you can track the thymidine. More killing means thymidine has been released, so then you can calculate how much killing has gone on. And we actually, looking at our lymphocytes against allogeneic and syngeneic tumor, there was no difference. And we thought maybe cytokine production is different, but it's not. Both in vitro and in vivo, again and again, looking at cytokines that may cause damage, they have been similar. So lastly, we thought, well, perhaps these knockout T cells are just unleashed. They travel faster for some reason. So what we did is we co-injected the wild type and knockout T cells. We isolated them, and then we stained them with different dyes so that we can follow them. And we co-injected them at a 1-to-1 ratio into radiated animals. And indeed, this is what we saw. So it may be hard to see the percentages, but at 24 hours in the same spleen, wild type is 26%, and knockout is about 70%. And at 48 hours, the wild type is about 20% and knockout 68%. And at 72, it's 10% versus 86%. And not only are they overtaking the wild type T cells, but these blobs, these peaks that you're seeing within the knockout population are division peaks. So these are dividing very early on. So to summarize, many of our patients still require transplantation, which is limited by graft-versus-host disease. Graft-versus-host disease is controlled by DNA methylation of our donor T cells, and that is mainly a CD8-driven process. Even though the wild type and knockout T cells in our experiments have similar killing and cytokine production, the knockout T cells move faster and divide early. And why is this important for our patients? Number one, we believe that the same way that the activated knockout T cells are better at causing graft-versus-host disease, they will be better at eradicating cancer. And in fact, there is recent published data that supports this. Researchers have gone back and have seen that if we use older donors that have asymptomatic DNMT3A mutations, using those donors for transplants results in lower risk of leukemia relapse in the patients. The second reason why this is important is because perhaps we can manipulate methylation. Perhaps we can induce donor T cell DNA hypermethylation to improve GVHD. And because we're We only have 15 minutes today. I'm not going to delve into these hypotheses anymore, but please come by our poster if you want to chat. Thank you to my mentors. Thank you to everyone who's helped in the lab. And most of all, thank you to our patients and families for inspiring us to work hard every single day.
Great. So are there some questions? I have a question I'll start with. So I'm just curious, the double positives that rose up during, and I'm not an immunologist, so I may get this completely screwed up. So if I do, just tell me I'm completely wrong. The double positives were present at a much higher percentage and evolved at a much higher percentage in the DMT3A knockouts.
Correct.
Double positives are representative of very early immune cells that have a certain level of then pluripotency. Okay, so have you been able to do or think about studies that now can dissect in a way that would lead to understanding T-cell ontogeny in a better way now that you know that you've got this model for studying it? Is there a way to understand T-cell ontogeny that will then, and obviously this may be your goal, to better increase the graft versus leukemia and, of course, decrease the graft versus host?
Right. So, to clarify, these T cells are not coming from immature T cells. These are coming from mature, naive T cells that we have given to our donors. So. This is too much to go back. So because we isolate bone marrow and T cells before the transplant, your bone marrow starts engrafting, but these T cells are not coming from the bone marrow. These aren't coming from sort of stem cell progenitors. These are coming from stem cells already. So it's not necessarily that something in their maturation, it's something in their activation that makes them re-express CD4. Now, what we can do about it is it is a large enough population that we can isolate it. So you can isolate, we have isolated by flow cytometry sorting, and we can do analyses to compare the wild type CD4, CD8, and the knockout CD4, CD8, and double positive. And you can do methylation analysis by whole genome bisulfite sequencing. You can do RNA sequencing. We have all sorts of exciting things lined up to take advantage of this very striking phenomenon in the knockout recipients to decipher what is different. How is it that methylation controls the T cells? And then, like you said, we can use the same exact mechanisms, the same exact analyses in GVL experiments. So you can do your transplant, and at the time of transplantation, inject a leukemia cell line that is syngeneic to your recipient. So it's going to grow out of control and only be controlled by your graft. And we can isolate the T cells after we have some leukemia controlled by the graft and do the same analyses, and even identify keys as to what is different between graft versus host disease and graft versus tumor activity.
No questions? Okay, well, thank you. Thank you.